In this chapter, you will learn to compare the functions of key transcription enzymes in eukaryotes and prokaryotes, such as RNA polymerases and transcription factors. You'll also analyze how transcription is regulated in eukaryotic cells through promoters, enhancers, silencers, activators, and epigenetic modifications. Unlike prokaryotes, eukaryotes use multiple RNA polymerases for nuclear transcription, at least 3. They also require dozens of accessory factors rather than just 1 or 2. Another key difference is compartmentalization. In eukaryotes, transcription occurs in the nucleus, while translation happens in the cytoplasm. Moreover, chromatin structure silences genes by default, making transcription highly regulated. Eukaryotic cells have multiple RNA polymerases with specialized roles. RNA pol synthesizes most ribosomal RNA. RNA pol 2 synthesizes messenger RNA. RNA pol 3 synthesizes tRNAs and some rRNAs. You can tell these polymerases apart by their sensitivity to toxins like alpha-amanitin. There are also RNA pol 4 and V in plants, involved in RNA silencing. Each of these enzymes contains 12 to 14 subunits, and mitochondria and chloroplasts have their own unique polymerases. Here's a key point. None of the nuclear RNA polymerases can initiate transcription by themselves. They all require help from transcription factors. Each polymerase works with its own specific set of transcription factors to initiate transcription at defined start sites. This dependency on accessory proteins distinguishes eukaryotic transcription from the simpler bacterial process. RNA polymerase II is responsible for synthesizing pre-mRNA and is structurally similar to taq-DNA polymerase, especially in how it handles template entry and product exit. It's composed of 12 subunits, making it a complex enzyme. We'll explore how this polymerase works with general transcription factors to initiate and elongate transcription, especially at protein-coding genes. RNA polymerase II is the enzyme primarily responsible for synthesizing messenger RNA-mRNA in eukaryotic cells. Structurally, it's similar to taq-DNA polymerase, particularly in the regions where DNA enters and RNA exits. This enzyme plays a central role in transcribing protein-coding genes, and its regulation is a major point of control for gene expression. RNA polymerase II is composed of 12 individual subunits, each contributing to its complex function. This architecture provides the flexibility and coordination needed to handle intricate tasks such as initiation, elongation, and termination. These subunits work in concert to interact with transcription factors, chromatin, and RNA processing enzymes. The core promoter spans about 40 base pairs upstream and downstream of the transcription start site TSS. It often contains a TATA box, a well-conserved DNA sequence located roughly 25 to 30 bases upstream. However, many promoters lack a TATA box and instead include alternative elements like the initiator inner, the downstream promoter element DPE, and BRE sequences that flank the TATA region. These motifs help define where transcription begins and how efficiently it proceeds. In human promoters, the initiator element is found in about 50% of genes. DPE and BRE elements are found in 25%, while TATA boxes appear in just 13% of promoters. This diversity reflects multiple modes of transcription initiation across the genome and highlights the modular architecture of core promoter elements. This review question asks, the -10/-35 boxes, TATA box, DPE, and BRE are all examples of what? The correct answer is C, promoters. These are specific DNA sequences that define where transcription by RNA polymerase begins. They are not mRNA or proteins, and though enhancers regulate transcription, they are not core promoter elements. In eukaryotes, RNA polymerase II requires the assistance of general transcription factors to initiate transcription. These include TFIA, TFIB, TFIID, TFIF, and TFI. Together, they assemble at the core promoter to form the pre-initiation complex. This minimal combination of factors and RNA Pol2 is sufficient for basal transcription, which is the low-level, unregulated expression of a gene. When a gene contains a TATA box, the general transcription process begins with the binding of TFIID, specifically its TATA binding protein TBP subunit, to the TATA box. This interaction bends the DNA, allowing the recruitment of other transcription factors, including TFIB, TFIF, TFI, and TFI, along with RNA polymerase II, completing the pre-initiation complex. TFIIH's helicase and kinase activities are essential for DNA unwinding and CTD phosphorylation, which trigger transcription initiation. Let's take a closer look at these transcription factors. TFIID with TBP recognizes the TATA box. TFIB helps position RNA polymerase II. TFIF stabilizes the complex and brings in Pol2. TFI recruits TFI, which performs two critical tasks: unwinding DNA and phosphorylating the carboxy terminal domain CTD of Pol2. This phosphorylation is a key step in allowing Pol2 to begin elongation. Not all promoters have TATA boxes. In TATA-less promoters, other core promoter elements like the initiator inner and downstream promoter element DP play a crucial role. In these cases, TAFs, TBP-associated factors within TFIID, recognize and bind these alternate sequences. The assembly of the pre-initiation complex continues in a similar fashion, enabling RNA Pol2 to initiate transcription even in the absence of a TATA box. Let's test your knowledge. Which of the following transcription factors is not part of the pre-initiation complex? The answer is D, TFIJ, because it doesn't exist. The other options, TFIA, TFIB, and TFI, are all legitimate components of the Pol2 transcriptional machinery. To transition from initiation to elongation, RNA polymerase II must undergo a key modification. Its C-terminal domain CTD, which contains repeated heptapeptides, must be phosphorylated. Specifically, serine 5 in the repeats is phosphorylated by TFI, allowing the polymerase to clear the promoter and begin elongation. Later, dephosphorylation is necessary for the polymerase to reassemble for the next round of transcription. While basal transcription machinery initiates transcription, it's usually inefficient. That's where regulatory promoters come in. These regions are located 50 to 200 base pairs upstream of the start site and often contain GC and CCAT boxes. Each gene has its own regulatory promoter, and maximum transcription occurs only when transcription activator proteins bind to these regions. Enhancers are DNA sequences that boost transcription. They can act from thousands of base pairs away and function regardless of orientation or position. In contrast, silencers repress transcription, often by altering chromatin or blocking activators. A related element, the upstream activating sequence UAS, is enhancer-like but must be upstream of the transcription start site. This slide visually reinforces that enhancers and silencers may act at a distance, and sometimes a single element can bind multiple regulatory proteins depending on cellular context. It sets the stage for understanding how DNA looping brings these distant regions into proximity with the core promoter.
For RNA polymerase to successfully bind to a eukaryotic promoter and initiate transcription, a set of proteins called transcription factors must first assemble on the promoter. The assembly process begins upstream from the transcription start site, where proteins called basal factors bind to a short TA-TA sequence in the promoter. Other basal factor proteins then bind, eventually forming a full transcription factor complex able to capture the RNA polymerase. Basal factors are essential for transcription, but cannot by themselves increase or decrease its rate. A second set of transcription factors, called coactivators, link the basal factors with a third set of transcription factor proteins called activators. Activators are regulatory proteins that bind to sequences on DNA called enhancers. Enhancers are located at sites that are distant from the promoter. The interaction of activator proteins with transcription factor subunits increases the rate of transcription. Many enhancers, scattered around the chromosome, can bind different activators, which provide a variety of responses to various signals. When a second kind of regulatory protein called a repressor binds to a silencer sequence located adjacent to or overlapping an enhancer sequence, the corresponding activator is no longer able to bind to the DNA.
Transcription activator proteins are crucial for recruiting the transcription machinery to a gene. These proteins typically contain two independent functional domains, a DNA binding domain that makes sequence-specific contact with regulatory promoters or enhancers, and an activation domain that interacts with the transcription complex to initiate transcription. Regulatory promoters and enhancers often share common DNA elements. The same transcription activator protein may bind both types. Whether a gene is expressed depends on a combinatorial process, integrating input from multiple activator proteins. This combinatorial control allows for precise regulation of gene activity. Transcription activator proteins, or TAPs, are categorized based on the structure of their DNA binding domains. There are four main families: helix-turn-helix, CY 4 zinc finger, basic region leucine zipper, ZN2CY6 binuclear cluster. The helix-turn-helix motif is found in Drosophila homeotic genes and vertebrate HOX genes. This domain has 3 alpha helices, and additional proteins often help refine binding specificity. It enables precise interaction with DNA to regulate developmental genes. This figure shows how homeotic genes in Drosophila are arranged in clusters. Each gene contains a helix-turn-helix DNA binding domain and is responsible for defining specific body segment identities during embryonic development. Found in the nuclear receptor superfamily, the CY4 zinc finger motif is a hallmark of transcription factors like the glucocorticoid, thyroid, and estrogen receptors. These proteins bind to DNA and hormone response elements HREs and play major roles in hormone-responsive gene regulation. The basic region leucine zipper domain is a fibrous, alpha helical structure. Every seventh amino acid is a leucine, enabling dimerization. Notable examples include the FOS and JUN oncogenes. These proteins bind DNA and regulate cell proliferation and survival. The ZN2CY6 binuclear cluster motif uses 2 zinc ions coordinated by 6 cysteine residues to stabilize DNA binding domains. Gal4, a transcription activator in yeast, is the best studied example. This domain is essential for activating genes involved in galactose metabolism. Activation domains are usually intrinsically disordered with no fixed 3D structure. 2 models explain how they work. In the recruitment model, they stabilize the transcription machinery. In the conformational change model, They trigger structural shifts that stimulate RNA polymerase II activity. In yeast, Gal4 is a model transcription activator with three key domains: a DNA binding domain spanning residues 1 to 64, a dimerization domain from residues 66 to 94, and a large activation domain from residues 95 to 881. This modular structure allows Gal4 to bind upstream activating sequences UAS and recruit transcription machinery only when appropriate, demonstrating the power of domain-specific regulation. The GAL1 gene is induced when galactose is present. Gal4 binds 4 UAS sequences upstream of the gene. However, repression occurs through MIG1, a transcriptional repressor that binds between the UAS and the transcription start site, blocking transcription in the presence of glucose. This dual regulation ensures yeast only express GAL1 when galactose is available and glucose is not. In the absence of both glucose and galactose, Gal4 still binds the UAS, but its activation is blocked by Gal80, an inhibitor. When galactose is added, Gal80 is removed, enabling Gal4 to activate transcription. This elegant switch demonstrates how protein-protein interactions regulate gene expression based on environmental signals. Glucose represses GAL1 expression by activating SNF1 kinase, which modulates the activity of MIG1. When glucose is high, MIG1 binds to its recognition site and blocks transcription initiation. This glucose repression system ensures energetic efficiency, allowing yeast to prioritize glucose over galactose metabolism. This figure dissects the functional regions of Gal4. It shows full-length Gal4, a peptide fragment containing residues 1 to 100. See the complementary residues 100 to 881 and D-Alexa-Gal4 hybrid used in functional studies. These experimental constructs reveal which regions are essential for DNA binding, dimerization, and activation, helping define the modular nature of transcription factors. The Gal gene system in yeast integrates signals from galactose, glucose, and intracellular proteins like Gal4, Gal80, and MIG1. It is a model for eukaryotic transcription regulation, showcasing how multiple transcription factors and co-regulators orchestrate precise gene control. This system exemplifies signal-dependent gene expression in response to nutrient availability.
This video will be on the second key difference between prokaryotic and eukaryotic gene regulation, and that is that eukaryotes tend to use very complex combinations of transcription factors to regulate individual genes. So this is in comparison to prokaryotes, which have these simple, maybe one repressor, one transcriptional activator. So there are three different kinds of regulatory elements that most eukaryotic genes have, and we're going to just mention these and then talk more about them. So first of all, the promoter. Okay, we have talked about this already, and we're not going to go into this in more depth, but this is the TATA box. This is where TBP binds and RNA polymerase will bind, and this is right next to the place where then transcription will begin, where the +1 site. Now there's something else called promoter proximal elements, and these are sequences that are fairly near to the promoter, and there are factors, proteins that bind to these sites and can assist in gene regulation. And then there are also enhancers, and enhancers are found at some distance from the start of transcription. And these act in the same way. They have transcriptional regulators, so transcription factor proteins that bind to them and can help to activate or repress transcription of that particular gene. So we'll talk about promoter proximal elements first. So promoter proximal elements are found within a short distance of the promoter of a gene, and usually within several hundred bases before the Tata box or the promoter. And so there are a number of promoter proximal elements that are pretty commonly found in genes. Two of those common ones are shown here, a GC box and a CAT box, as they're called. And to these particular sequences, proteins will bind, and they can be important in activating transcription. And so what you can see here, this is just measuring the levels of transcription of this particular gene. And if you mutate, of course, the Tata box, all right, you lose transcription, but also if you mutate the CAT and the GC boxes, you also lose that those high levels of transcription. So most eukaryotic genes have at least some promoter proximal elements. There are a number of different ones in addition to these two that I've mentioned here. However, they all are defined by their proximity to the normal promoter. Enhancers, on the other hand, can be quite a distance away from the gene. And so they can be before the gene or after the gene, or they have even been found within the gene in an intron. So to these particular enhancer sequences, transcriptional regulators will bind, and these can help then to activate or to repress transcription of that gene. We'll talk about two specific examples of how individual genes are regulated in eukaryotes. The first example will be the GAL4 system in yeast. This is a relatively simple example, and it's meant to be analogous to the lac operon in bacteria so that we can compare and contrast the differences between prokaryotes and eukaryotes. The second example is rather complex, the human beta interferon gene. We'll talk about this in class. It won't be on this video. Both of these examples are going to illustrate how we use transcription factors, but then how these transcription factors tie back into this whole thing of regulation of epigenetic marks in histones and methylation of the DNA. So the first example is the GAL system in yeast. And so remember, yeast is a unicellular organism, one of the most simple eukaryotic organisms. And the idea here is that yeast need an energy source, and their preferred energy source is glucose, right? But they can also use other sugars, for example, galactose. And so galactose needs to be converted into glucose 1-phosphate in order to be able to be used in glycolysis. And there are a number of genes that are required to do this conversion. Okay, so galactose is up here, and there's a number of different steps in the conversion of galactose into glucose 1-phosphate. These steps are catalyzed by these GAL genes, such as the GAL2 gene, right, the GAL1 gene, GAL7, and GAL10 gene. Now, just like in bacteria, we want these genes only to be transcribed when galactose is present. So only when this energy source is present should we bother to make all of these proteins that are required for the metabolism of galactose. And so the way this works is the following. So there is a transcriptional activator called GAL4, and GAL4 will bind to particular enhancer elements near to these GAL genes. So these 4 GAL genes are similar to those structural genes in the lac operon, all right? And so these are the ones that need to get activated. When GAL4 binds to these enhancer elements, so these enhancer elements are called upstream activating sequences, okay, or UAS sequences. When GAL4 binds, then the nearby GAL gene is transcriptionally activated and gets transcribed and then translated at high levels. Just want to point out a little bit more about this particular GAL4 protein. Okay, so this is actually a dimer. There are 2 GAL4 proteins that bind together, and there are two primary, very important parts to this. So one part is the DNA binding domain, and the other part is the activation domain. All right. There's also the place, of course, that allows these two proteins to bind together. That would be called the dimerization domain. So here's how this works. First of all, let's keep in mind the big picture. So when we do not have galactose present, okay, that's shown on the top here, we don't want to transcribe the gal genes, all right? In contrast, when galactose is present, we do want to transcribe the gal genes. Now, the way things work is a little bit more complicated than this very simple case we saw in prokaryotes, where the repressor was the thing that responded to that effector. So here you've got a much more complex situation. You've got a couple of other proteins that are very important in regulating the activity of gal4. And all three of these proteins, so gal4, this protein gal80, and gal3 are always present in the cell. Okay, but they will be able to respond to the presence of galactose in the following way. So normally, if you don't have galactose present, we have gal4 is present in the cell and it will bind to that enhancer. However, it is inhibited in the activation domain by another protein called gal80. Okay, so gal80 will just sit, has a very high affinity for that gal4 protein, and will sit on it and prevent it from activating those nearby gal genes. Now, the protein which actually binds to galactose when galactose gets added here is this protein gal3. Okay, so gal3 is just hanging around doing nothing in the absence of galactose, but once galactose is present, galactose or gal3 will bind that galactose, and that changes the conformation of gal3, and what it does is it highly increases the affinity of gal3 for this repressor protein gal80. Okay, and so what happens is that gal3 now has a higher affinity for gal80 than does gal4. And so gal80 gets bound to gal3 and gets pulled off of that gal4 transcriptional regulator or transcriptional activator. Okay, this is now free to activate those nearby gal genes. So what does the gal4 activation domain actually do in order to activate transcription? So some of this is very similar to a transcriptional activator in prokaryotes. So remember, our transcriptional activator in the lac operon was CAP, and that acted to help recruit RNA polymerase to the promoter. And so gal4 does some similar things here. So first of all, it binds to TF2D complex. Remember, TF2D is one of those general transcription factors that must bind to the promoter. TBP is part of the TF2D complex, and that gal4 binds to this and helps it to bind to the promoter. Another thing that the gal4 activation domain does is it binds to a complex of proteins called the mediator complex. Now, we haven't talked about this before, but what the mediator complex does is it binds to RNA polymerase and helps it bind onto the promoter. All right, and so those first two functions here are very similar to what happens in prokaryotes. We need to get all of those general transcription factors and the RNA polymerase bound to the promoter. The third thing is a little bit different, however. So the other thing that gal4 does is that it recruits the SY-SNF complex to this chromosomal region. So SY-SNF, remember, moves nucleosomes around. It remodels the nucleosomes, and it can help to move nucleosomes that are sitting right on that promoter. It can move them away, allowing the binding of these general transcription factors and RNA polymerase. Now, there's a completely additional way that the gal genes are regulated in addition to the gal4 system. So just like the lac operon, when glucose is present, the cell would prefer to use glucose rather than galactose. So just like with lac, it preferred glucose instead of lactose. The way this is accomplished is via a protein called MIG-1. Now MIG-1 gets higher when the glucose levels are high in the cell, and MIG-1 will bind to another enhancer site called the MIG-1 site, all right, near each of those GAL genes. When MIG-1 binds to the DNA here, it will recruit a second protein called TUP1. Now TUP1 is called a co-repressor, and it has a certain enzymatic activity. It is a histone deacetylase. All right. Now, if you remember, histone deacetylases remove those acetyl groups from the nucleosomes, and the result of that is that the chromatin is more tightly packed. We can't move those nucleosomes around as well, and you tend to have inactive genes in that region. All right, and so when glucose is high, MIG-1 binds, and MIG-1 can actually, act on those nucleosomes dominantly over the effect of GAL4, right? And so you're going to turn off those GAL genes when MIG-1 is present, and the nucleosomes become deacetylated.
The mediator complex is essential for activated transcription. It's a multi-subunit assembly with 3 core domains. The head contacts the C-terminal domain CTD of RNA pol II, the middle bridges communication, and the tail receives signals from gene-specific activators. Mediator acts as a molecular bridge, conveying regulatory signals to the transcription machinery and enabling context-specific gene expression. Chromatin restricts access to DNA, and RNA pol II cannot function on nucleosome-bound regions. To address this, eukaryotic cells utilize ATP-dependent chromatin remodeling complexes. These powerful machines reposition, eject, or unwrap nucleosomes, allowing transcriptional machinery to access gene promoters and regulatory sequences. This figure shows the versatile activities of chromatin remodelers. These complexes can slide nucleosomes along DNA, eject them completely, or displace histone dimers, making the DNA accessible for transcription. These remodeling actions are essential for gene activation, especially in tightly packed chromatin regions. Epigenetic regulation goes beyond DNA sequence. Histone acetylation, mediated by HAT's histone acetyltransferases, loosens chromatin and promotes transcription. Conversely, HDAC's histone deacetylases remove these marks, leading to gene silencing. These modifications are heritable, reversible, and critical in development and disease, demonstrating that transcriptional regulation is not just about sequence, but structure and context. In addition to acetylation, histone methylation is a key epigenetic mark that regulates transcription. Depending on the site and number of methyl groups, this modification can either activate or repress gene expression. For example, H3K4 methylation is associated with gene activation, while H3K9 or H3K27 methylation typically indicates repression. These modifications recruit reader proteins that establish distinct chromatin environments, adding yet another layer to transcriptional control. This schematic shows histone modifications mapped across the core histone proteins H2A, H2B, H3, and H4 within a nucleosome. Each circle represents a specific chemical modification. Red circles for acetylated lysine, white circles for methylated arginine, green circles for methylated lysine, blue squares for phosphorylated serine. Key residues like H3K79 and H2BK123 are also highlighted, emphasizing how different marks on different tails collectively form the histone code. These modifications guide transcription, DNA repair, and chromatin compaction by recruiting specific reader proteins. This animation depicts the action of histone acetyltransferases, or HATs. These enzymes acetylate lysine residues on the histone tails, neutralizing their positive charge. The result is relaxed chromatin, allowing transcriptional machinery to access DNA more easily. Histone acetylation is a hallmark of active gene expression and plays a key role in development, memory, and disease.
Epigenetics refers to the heritable modifications in gene function or activity without changes in the DNA sequence. Epigenetic processes are genetic modifications that affect gene regulation by changing the DNA conformation. Epigenetic changes are found in many different cancer cell types. One such change is histone modification that may be caused by overexpression or aberrant recruitment of HDACs. Within the chromosome, DNA is packaged into chromatin. Chromatin consists of DNA, structural histone proteins, and non-histone proteins. Within chromatin, the repeating unit is the nucleosome. Nucleosomes are made up of 146 base pairs of 2 superhelical turns of DNA wrapped around a core of 8 histones. The histones are responsible for maintaining the chromatin's shape and structure. Epigenetic modifications, such as histone acetylation, occur at the amino terminal tails of the histones that protrude from the nucleosomes. Acetylation of histones is generally acknowledged to play a key role in the regulation of gene expression. Histone acetylation is controlled by the balance in the activity of 2 enzymes, histone acetyltransferase, or HAT, and histone deacetylase, or HDAC. For a gene to be transcribed, it must become physically accessible to transcriptional machinery. Acetylation of histones by HAT causes uncoiling of DNA and an open chromatin structure. This causes genes to become accessible to transcription factors, allowing gene expression to occur and proteins to be made. Conversely, deacetylation of histones by HDAC results in tight coiling of the DNA and a closed chromatin structure. In some cancer cells, there is an overexpression of HDACs, an aberrant recruitment of HDACs, or an underexpression of HATs, resulting in hypoacetylation of histones and therefore a condensed or closed chromatin structure.
RNA polymerase I is responsible for synthesizing most of the ribosomal RNA, specifically the 5.8S, 18S, and 28S rRNA components. These RNAs are synthesized in the nucleolus, where poly operates. Note that 5S rRNA is transcribed separately by RNA polymerase III. Poly is structurally similar to Pol II, but it is highly specialized for high-output rRNA transcription, which is critical for building ribosomes in growing cells. This diagram compares pre-rRNA transcription units in human, mouse, and frog xenopus. Each unit contains sequences for 18S, 5.8S, and 28S rRNA, interspersed with internal transcribe spacers, ITS1 and ITS2, and flanked by external transcribe spacers, or ETS. These spacers are removed during processing to form the mature rRNAs. Although species vary slightly in length, the overall structure and order are highly conserved, reflecting the essential role of rRNA in protein synthesis. This figure shows the structure of the rRNA transcription unit, which includes a core promoter, where transcription begins, and an upstream promoter element UPE that enhances initiation. Together, they control transcription of the 18S, 5.8S, and 28S rRNA genes, highlighted in green, cyan, and red respectively. These elements are essential for efficient transcription by RNA polymerase I, especially in the nucleolus, where ribosome biogenesis occurs. RNA polymerase III synthesizes short, structured RNA molecules vital to the cell's function. Two key products are 5-srRNA, a component of the large ribosomal subunit, and tRNAs, which carry amino acids during translation. A single eukaryotic cell can have 30 to 100 different types of tRNAs, emphasizing the complexity and importance of Pol3 activity in supporting protein synthesis. RNA polymerase III uses 3 different promoter types to initiate transcription. Type 1 promoters, for 5-srRNA, are located within the transcription unit. Type 2 promoters, used for most tRNA genes, are also internal. Type 3 promoters, for small nuclear RNA centernas, are found entirely upstream of the transcription start site. This structural diversity allows Pol3 to control transcription across a wide range of small functional RNAs. This figure organizes the 3 promoter types for RNA polymerase III. Figure A shows the type 1 promoter layout for 5-srRNA genes, with internal elements located downstream of the start site. Figure B depicts the type 2 promoter used in tRNA transcription, featuring box A and box B motifs inside the gene. Figure C illustrates the type 3 promoter, found upstream in centerna genes, resembling typical Pol2-like architecture. These structural differences reflect how RNA Pol3 adapts its initiation strategy to the type and function of the target RNA.